CCPP, its prevention and control. There are so many respiratory diseases that encountered with the sheep and goat. They are confusing with the clinical sign. Broadly, we can categorize these respiratory diseases of sheep and goat in two categories. One is upper respiratory tract disease like nasal polyp, rhinitis, granuloma, tumors, or pharyngeal and laryngeal paralysis. And other one is the disease of lower respiratory tract like tuberculosis, CCPP, pasteurellosis, and hemorrhagic septicemia. Out of them, the contagious caprine pleuropneumonia, that is the CCPP, is a very important disease. First of all, we have to discuss about its background. By understanding the background of this disease, we can understand the present situation of the disease. This is the highly infectious and contagious disease, particularly of goat. And less commonly, is this disease is also occur in the sheep and other wild ruminants. The disease is characterized by the unilaterally serofibrinous pleuropneumonia. Now the disease is found in more than 38 countries. These countries are mostly lies in the Africa, Asia, and Europe. This disease posing the severe socioeconomic importance. The sheep and goat rearing system is mostly present in the lower income group countries, or we can say in developing countries, where the poor farmers are mainly depend on the sheep and goat for their livelihood. So this disease is of highly infectious and contagious nature and causing the heavy mortality. So it poses the negative impact on their livelihood. Not only the economic conditions of the farmers, but it also affects their culture, their trade and nutrition, because the sheep and goat both are reared for the nutrition also. They are getting the meat from these animals and also getting the milk from these animals. If the animals suffered with this disease, then nutrition is also compromised. This disease also affecting the international trade, and the OIE listed this disease as a notifiable disease. The OIE listed this disease in list B, where all those diseases were put together. Those diseases causing the socio-economic losses within a country. So the trade is not only hampered internationally, but also hampered nationally. In an estimation, this conducted by the Yatu and co-workers, that this disease posing the burden globally, which is estimated at the US dollar 507 billion per annum. This disease is caused by the Mycoplasma capricolum subspecies capri pneumoniae. In short, we can say it is the MCCP. Previously, this organism is recognized as, and called as Mycoplasma biotype F38. This organism is difficult to grow in the laboratory because the organism requires a specialized media, and the incubation period in the laboratory is also very long. So it is very much difficult to grow. When it is grown, then the colony have particular characters that the colony appears the fried egg like, and the centrally the rose colored appears due to the reduction of the triphenyl tetrazolium salt that is being used for the growth of this organism in agar. This organism is the part of the Mycoplasma mycotis cluster, where some other organism also put together. That are the Mycoplasma capricolum subspecies capricolum, Mycoplasma dg, Mycoplasma mycotis subspecies capris, Mycoplasma mycotis and subspecies mycotis. These different subspecies can be identified or differentiated by only highly specific PCR test that requires a specialized laboratory. This is the fastest evolving organism that have the high mutation rate. So it is very difficult to produce a effective vaccine against this organism. This organism only having the some degree of heterogeneity that commonly not found in the members of Mycoplasma mycotis cluster. Based on the sequencing of the several genes, including 16S rRNA and H2 locus and other protein, 24 haplotypes were identified in 25 strains of Mycoplasma capricolum, subspecies capricolum, and placed within 6 genotyping groups from A to F, with 2 different evolutionary lineages. Lineage 1 contains 2 groups with the strains from East Africa, Qatar, Nigeria, and Mauritius. Lineage 2 is subdivided into 3 groups with the strain from United Arab Emirates, China, and Tajikistan. If we talk about the other properties of the Mycoplasma, that this organism is a cell wall-less organism, and it requires the sterol. It is catalase and negative, non-motile, and pleomorphic. The shape of this organism is varying. It is from spherical, ovoid, or pear shape, and having branched filamentous form. This organism is very much fragile and unstable in the external environment. This can be up to 3 days in the tropical zones, and it can survive in up to 2 weeks in the temperate zones. This can be destroyed in the environment at the temperature of 56 degrees centigrade for 60 minutes. If we increase the temperature up to the 60 degrees centigrade, it can inactivate it within 2 minutes. But if we keep this organism in the frozen condition, then it can survive up to the 10 years. This organism can also be destroyed by the formaldehyde and the mercury chloride at their particular concentration for a particular time. The routine disinfectant which we are using in our daily work, that are also effective. Like 1% phenol can destroy this organism within 3 minutes. This is the host-specific organism. It mostly affects the goat very seriously, but sheep can be infected. Sheep can also be infected experimentally. The captive and free-ranging wild animals, including deer, gazelle, and ibex, are affected, and the organism have been isolated from these species. The morbidity rate is very high, that is 90%, and case fatality rate also very high, that is from 60% to 100%. So we can assume that how much the disease is deadly for the animals, and how can this disease create havoc among the farmers. The incubation period of this disease is 6 to 10 days, but it can be up to the 28 days. The disease does not having any genetic importance, so it is not a matter of concern for the human being. If we see about the prevalence rate of this disease, we can see the trend of CCPP over the all over the globe, that the blue dots is indicating the prevalence trend in the sheep over the year of from 1979 to 2018, and the goats are denoted by the red dots. So here is the trend of the prevalence. This prevalence trend is based on the several studies which have been included for 41 studies. The present scene of this disease in our country is highly variable. The prevalence rate is from 5% to 64%. That is reported by different researchers. The disease in our countries have been reported by many states like Tamil Nadu, Maharashtra, Gujarat, Kerala, Himachal Pradesh, Jammu Kashmir, Rajasthan, by different workers. So this disease is now not a new disease in our country, and it also some sporadic cases have been reported in different part of different states. Predisposing factors of this disease are poor sanitation. We are knowing that our farmers are not having a very good sheds for the animals. The design of the sheds and their housing is not so much proper that they have the good sanitation conditions at their animal houses. So the poor sanitation condition provoke this disease. One factor is the overcrowding, because the goats the open grazing system requires more spaces, and if the number of animals is very high in the stall fed animals, so there is also a requirement of more land. So overcrowding is generally happening in our country. So it plays important role in getting the infection from infected animal to the healthy animals. If the housing is not proper, there will be a poor ventilation. This can also prompt the disease. Excessive dust, variation in the environmental conditions like higher temperature, lower temperature, higher humidity, higher current, air currents. These factors causing the stress among the animals, and when the animal comes under the stress condition, their immunity will be compromised. Due to the compromised immunity, there is the chances of flaring of this disease in the animals. Other various kind of stresses are also there, like pregnancy stress, lactation stress, or nutritional stress. There the animal are not getting the proper feeding. These stresses also provoking the disease. If we talk about the zerograzing system, then there is the close contact of the animals in a particular place, and that causing the poor hygienic condition, and the animal remain in close contact, and so the chances of getting the infection from the infected animal to the healthy animals increases to the animals. In grazing animals, particularly in case of sheep, we are seeing that the animals keeping their head close to each other, and the secretions of the respiratory system, they can come in contact with that secretions, and if the secretions having the organism of CCPP, then they can get the infection. The transmission of this disease, as we know that the disease is highly contagious and infectious in nature, so it can spread rapidly by the close contact. The main mode of the transmission is the aerosol droplets. If the aerosol droplets, these are formed due to the sneezing and coughing by the infected animals, and if the animal is suffered with the CCPP, and then the organism may be secreted or in the sneezing and coughing in the form of a droplet. If these droplets containing the organism and inhaled by the healthy animals, then there is a chances of getting the infection. The respiratory secretions that is purulent to mucopurulent or serous discharges of the infected animal. If the healthy animal comes in the contact, then it can get the infection. The formats also have the way of transmission. The formats like fur, hairs, the handler's cloth, handler's hand, metal bars, and walls, and premises where the animal are being kept. These can be infected with the secretion of the infected animals, and when healthy animals come in contact with these secretion, they can get the infection. In infected area, the major source of infection are with the carrier animals. The carrier animals always remains a source of infection. They are continuing shedding the organism in their respiratory secretions. This organism do not having any predilection towards the age groups and the sexes. So the disease can be occurred in any age of the animal, or in both male and female. The clinical findings noticed in the animals, particularly the clinical signs, are restricted to the respiratory system only. No other system is majorly affected. Few clinical signs may be observed as a arthritis. The affected animal may show high grade of fever, ranging from 104 degree Fahrenheit to 106 degree Fahrenheit. The animal usually coughing, and if the disease is progressed, then the coughing also be painful. There will be dyspnea, and if the severity is higher, then the animal extends its neck. Breathing is rapid and shallow, and there will be generalized weakness in affected animal. If we see the first picture, then the goat is showing extended neck due to the severe dyspnea, and goat is keeping its forelimbs wide apart. The second picture is showing that there is the mucopurulent nasal discharge and having the painful coughing. Next picture is showing the conjunctivitis, and as the disease is progresses, the animal showing the reluctance to move, and they oftenly laying down, and but they are remain on usual feeding, and they can stand and walk properly. But in case of terminal stages, respiration rate increases very fast. There will be open mouth breathing. Tongue may be protruded. There may be chances of frothy salivation. Animal keeps its elbow abducted, and animal may die within 2 or more days. The disease in kids comes in severe form, that is the septicemia form, and the kids may be suffered very badly. We all know that the Mycoplasma also causing the mastitis in lactating goats can cause egg-electic conditions, that there is complete loss of milk from the lactating animal. The clinical findings we can broadly categorize in 3 forms. In percute form, sudden death of animals without showing much clinical signs, except some extent of dyspnea. But in case of acute form, there will be high fever, coughing, dyspnea, labored breathing, excessive nasal discharges, that is from mucopurulent to purulent, corneal opacity, anorexia, depression, animal disinclined to move, and animal may die within 1 to 2 weeks. If we talk about the chronic condition, there will be no febrile condition. Coughing may be there for a longer period. The animal is emaciated and showing the stunt growth. The animal does not showing any response toward the given treatment, and death may be occurred after the 30 to 40 days. Rehman and co-worker recorded the percentage of clinical signs. They reported that the pyrexia, coughing, pneumonia, and nasal discharges showing by the animals in 50%, more than 50% cases. And prevalent clinical sign remains is the nasal discharges. That is almost 60% animal shows the nasal discharges. And least clinical sign that were observed by these researchers is polyoarthritis, only in 2.5% animals. The clinical sign together seen in animals, that is the only 2.5%. The necropsy finding is very important clinical important finding for the diagnosis of this disease, as the clinical signs that are restricted to the lungs and pleura. Similarly, the necropsy findings are also restricted to the lungs and pleura. In first picture, you can see there is the congestion of the lungs, and this part become consolidated, and this is called the red hepatization. In the second picture, the lungs become completely consolidated. In this picture, we clearly see the reddish area that is being consolidated, and this is the congested part. Collectively, we can say this is the red hepatization. We can also see here is the extracellular fluid that is filled in the pleural cavity. This picture here is the serofibrinous pleuritis that involving the lungs. In this picture, we can see the extracellular, or it is fluid in the pleural cavity, and in this picture photograph, we can see the interstitial pneumonia. This is a very important photograph. In this slide, we are seeing that this part is called sequestrum. Like in other photograph, this is also called sequestrum. Sequestrum is the dissociated part of the lungs, which is completely separated from other part of the lungs. Here there is no blood supply, or that is not involving in the respiration process. This sequestrum is formed due to the accumulation of the infective organism in a particular area, and it remains as a capsule, although it does not causing much problem to the animal. But in certain predisposing conditions, and if this capsule is burst, then the infection released from this area, and the infection disseminated to the other part, particularly affecting the respiratory tract, and the animal become source of infection. This photograph is showing the fibrinopurulent part, and here is the adhesions have been we can observe adhesions that is attached to the intercostal muscles of the thoracic cavity. These are the adhesions, and this is the lungs. Lungs is become congested, and some necrotic part you can also see. Here is the accumulation of extracellular fluid in the thoracic cavity. In this photograph, here is the completely consolidated lung part. You can see here is the some necrosed part, and the whole lungs become congested. Here we can also see the marbling of the lungs. The interlobar septa become widened, so it looks like the marbling. This is showing the pericarditis, and also this is the lungs showing the marbling. Here is the congestion of the pericardium. The fibrosis in the lungs, and here it is the infiltration of the leukocytes that we can see in the blue dots, and here is the pulmonary emphysema. The alveoli become widened, and it is flooded with the fluid. In this photograph, we can also see these conditions: infiltration of the leukocytes and pulmonary emphysema. This photograph is also showing the intra-alveolar capillaries that is flooded with the alveolar spaces, the bronchioles. The second photograph is the magnification of the first photograph that is showing the fibrinous and inflammatory exudate within the bronchiolar lumen. For the diagnosis, we can depend on the visible clinical signs, necropsy findings. The necropsy finding and clinical signs indicating towards the disease. The epidemiological data of the particular area can also help us for the diagnosis. If we analyze the hemogram, then we can see there is the increase in the WBC count, and there will be thrombocytopenia. There are not much alterations in the serum biochemistry. Only you can find the decrease in total protein concentration. And the other way for the diagnosis is the real-time PCR, and some serological test like complement fixation test, competitive ELISA, latest agglutination test, and heme agglutination test will help in the diagnosis. The latest agglutination test is a screening test for the CCPP. It is used in the screening of the disease. The other way for the confirmatory diagnosis is the cultural examination. For the cultural examination, we have to take samples from the lesions of from the lungs. Particularly, we have to take the samples from the where the consolidated and unconsolidated area is made. We can also take samples from the pleural fluid and mediastinal lymph node. As we talk about the cultural property of this organism, that it is very difficult to culture in the laboratory. So because it requires a specific media for this organism, media generally used is the Van de Foy goat media, WJ media, modified Haflick media, modified Nevin streptose. Another drawback for the culture of this organism is that it's require a longer incubation period, incubator, that is around 7 days or even more. For the molecular identification of this organism, the PCR assay is a choice of technique for the confirmatory diagnosis of CCPP. Two PCR assay nowadays are popular that are based on different gene. The amplification of 16S rRNA gene, or another one assay is the amplification of MCCP SPA FR gene. The differential diagnosis for this disease, although there are so many respiratory diseases which are confusing the CCPP. It is very difficult to differentiate this disease in the field condition. The other organism of the Mycoplasma mycotis cluster confusing this disease. Another one is the PPR. PPR also showing the clinical sign of respiratory system, but we can differentiate clinically this disease from CCPP as the PPR also having the diarrhea and the erosion of mucosa in the buccal cavity. So we can easily differentiate this disease. It is difficult to differentiate pasteurellosis from this disease because pasteurellosis also having the clinical signs that are limited to the respiratory system. This can be only differentiated in the laboratory examination. For the treatment of this disease, the antibiotic therapy have been recommended by several researchers. That is the tylosin at the dose rate of 10 milligram per kilogram body weight BID for consecutive 3 days, and denofloxacin at the dose rate of 2.5 milligram per kilogram per day for 3 days. The oxytetracycline in higher doses, that is 15 milligram per kilogram per day, is also effective, and this medicine is widely used in case of sheep and goat. The oxytetracycline is very much popular among the farmers, and they are taking this medicine from the medicinal counter, and they are using their by themselves. The long-acting oxytetracycline at the 20 milligram per kilogram body weight as a single shot is also effective in this condition. Nowadays, tilmicocin as a single shot, 15 to 20 milligram per kilogram body weight is also being used. Marbofloxacin at the dose rate of 2 milligram per kilogram body weight for 3 to 5 days by any means of route, IV, IM, or subcut route is effective. This medicine can also be given as a single shot at the dose rate of 8 milligram per kilogram body weight. But the antibiotic therapy is generally recommended with a proper strategy to control this disease. The sheep and goat are the major source of protein for the from the animal origin for human being consumption. So the antibiotic residues in the meat and milk of these animals posing a great concern of antibiotic resistance in the human population. So the indiscriminate use of the antibiotics in the food animals having the great concern all over the world. So it should be used judiciously and in a proper strategy to contain this disease. If we treat the animal properly, then animal may survive. But the treated animal may become the carrier, and they are becoming the source of infection. For the prevention and control of this disease, there are several measures have been suggested by the OIE. This disease is under the notifiable disease that we have. If anybody has noticed or this disease, he must have to report this disease to the higher authorities. That by that means the higher authorities can make a proper strategy to contain this disease in a particular area, and they can get success in prevention and control. Effective biosecurity measures should also be adopted as a isolation and quarantine of the animals. In countries like our India, there are less emphasis has been given on the biosecurity measures. Introduction of new animals in previous herd, or introduction of newcomer in the herd for visiting purpose or other any educational purpose. There are three to four policy strategies commonly used for the eradicating of this disease. One is the eradication by the slaughter of all infected and exposed animal. Practically, this policy is opposed by the farmers and practically not possible. Uh, Other one is the eradication by the quarantine, testing, and slaughter policy. Uh, Other one is the testing of the animals and strict imposition of the rules for the movement of the animals. So there will be no migration of the animal from the infected area to the other areas, or other animals to the infected area. There is complete ban on the animal purchasing or selling from that particular area, and there will not be any fair, animal fair. Another widely accepted policy is to vaccinate the animals. The live attenuated vaccine have been trialed, but these vaccines are commercially not available. Another one is the immunization with the killed vaccine, but these vaccines requires a booster dose after 1 month, because these producing the shorter immunity. The available vaccines are the bacterial vaccines. For the production of these vaccines, OIE provides the guidelines. The vaccine developed in the 80s and 90s. The vaccine is administered as the early as 4 months of age. It requires the revaccination in every 6 months, because it produces the immunity for a shorter period. The vaccination also having some limitation besides some benefits. The vaccine of the CCPP is expensive in comparison to the other goat vaccines. These vaccine can cause inflammatory reaction at the site of inoculation, as we are seeing in case of FMD in the large animal. These provide the immunity for a shorter duration, only for 6 months. But these vaccines, unfortunately, not available in India, as no vaccine manufacturer is coming forward to manufacture the CCPP vaccine.